A positive temperature change acts as a strong stress stimulus, and that has to be first realized by certain macromolecules that are employed by the cell. Now these heat sensing mechanisms, they can either be direct, where the temperature directly affects either the configuration or the activity of the sensor, or they may be indirect, where the changes occurring because of the stimulus, that is rise in temperature, act as a secondary stimulus, and then they are detected by certain other macromolecules. So taking temperature as a primary stimulus, the microbes, the plants and animals, they have certain thermal sensors, and these can be DNA, RNA as well as proteins. Now talking of the RNA acting as sensors is a very common phenomena in microbes, and these structures are then called RNA thermometers. So they were first discovered in E. coli, and most of the studies have been done in response to expression of sigma 32 factors, that's a heat shock alternative factor. Within the RNA thermometers, there are certain regions which can form secondary loops. So when these secondary loops are formed, there is intramolecular base pairing. So this 4U residues, these play very important role, and they have their corresponding bases at some other region within the RNAs with which they can base pair. So these regions, they are generally found in the 5' UTRs. So they may include promoters as well as, under some exceptions like the sigma 32 factor gene RPOH, they also include the coding sequences. Loop formation, obviously the ribosomes cannot access the ribosome binding site, that is the Shine-Delgado sequence. So in that case, the translation of sigma 32 is not permitted. So these things, they are occurring as the non-permissive temperature. So under normal conditions, when the temperatures are optimum, the translation of sigma 32 protein, the transcription factor, is restricted. As soon as the temperatures increase, the energy of the atoms increases, the van der Waal interactions and all such associations, they get broken up, so that the secondary loop starts opening up. With further rise in temperature, when the loop is completely opened up, now the Shine-Delgado sequence is accessible to the ribosome. So when these ribosomes bind, further translation occurs, and these which can go on to release the repression and gene expression occurs. Similarly, there are DNAs which can also act as thermosensors, and in those sense they are called DNA thermometers also. Due to rise in temperature, the molecular membranes, be it cell membranes or be it organellar membranes, all these get affected. So when mitochondrial membranes get affected, the ATP generation machinery is repressed, and the ratio of ATP ADP gets lessened down. So there is less of ATP in the cell. Now the enzymes which are dependent on ATP, their function gets modulated. One such important enzyme is DNA gyrase. So this gyrase is known for its supercoiling activity. So gyrase is the one that helps remaining DNA in a particular configuration, in a particular conformation state, and this enzyme needs ATP. In presence of ADP, gyrase gets inhibited. Now its correlation with the activity of the DNA is, when the promoter regions are involved here, then they are highly supercoiled, so RNA polymerase is not going to bind there, so the expression of that particular gene is restricted. It has to open up in response to the change, which is high temperature stimulus here, and then only expression begins. So most of the time this DNA thermometer thing occurs with the regions that are either involving promoters or near the promoters, so that at non-permissive temperature the transcription won't occur, and at permissive temperatures the transcription gets enhanced. Another example where DNAs can act as sensors of temperature change is where local DNA curvatures affect the binding of regulatory proteins which can cause changes in the expression pattern. One such example is RSE like VRF regulator. So VRF are proteins which are formed by pathogens, and when they thrive outside the host body, that is at the non-permissive temperature, the formation of mRNAs related to this gene, those are not done, and when they enter inside the host, experience a rise in temperature, then at that permissive temperature the relevant genes are expressed. So at the temperature of nearly 28 degrees Celsius, there is a particular loop formation in the promoter near the VRF gene, which provides a binding site for a repressor, that is nucleoid associated histone like protein called HNS. When there is rise in temperature, the van der Waals interactions, they get disrupted. So this particular loop near the promoter, it opens up, and now these HNS, they cannot bind at their related sites. So under those conditions, now RNA polymerase is allowed to bind and the expression of genes occur. So here we see a de-repression occurring in response to rise in temperature because of opening up of DNA loop. So in this way the DNAs, they can act as direct sensors of change in temperature. Besides DNA and RNA, proteins play a very major role acting as sensors. So there are different classes of proteins that can act as sensors. There are kinases, there are chaperones, repressors, proteases, and as per the definition of sensor molecules, these proteins, they undergo a conformational change in response to the stimulus, which is the rise in temperature, and this conformational change, this can bring about downstream changes, such as if there is a conformational change in repressor, now it won't be able to bind at the operator site and the repression will be lifted. Now an example of this kind of regulation whereby proteins are acting as sensors is very clearly evident in the activity of heat shock responsive transcription factor, that is sigma 32. As evident, there is a direct physical interaction of these chaperones, namely DNA K, J, GRPE, and also this is supported by chaperonin GRO-EL. So these basically keep a negative control, that is they limit the amount of sigma 32 within the cell. Now this sigma 32, it also gets helped up by SRP, that is signal recognition particle, and it is taken up to this SRP receptor which is localized in the membrane. So once it is localized in the membrane, it is also bound up by FTSH, which is a protease, and this protease helps in degradation of sigma 32. Now there are two aspects to it. First, the membrane localization of sigma 32, it enhances its capability to visualize the changes occurring in the membrane proteins, again which can act as sensors, and also the intracellular protein status, that is folding and unfolding thing. And also, since FTSH is acting as a protease, when there is change in activity of FTSH, then the level of sigma 32 gets changed. So this is how temperature is acting upon, because under change in temperature towards a positive side, the rise in temperature, that causes disruption in this FTSH activity, and therefore the degradation of sigma 32 gets reduced. So there is buildup of this transcription factor. On the other side, since temperatures also denature proteins, so these chaperones and chaperonins, which are at first attending to the function of reducing the amount of sigma 32, they now get channelized to these denatured proteins. They start attending to these denatured proteins, and therefore this negative feedback loop gets lifted. So overall, a rise in temperature through degradation of FTSH, through engagement of chaperones and chaperonins, this will allow the amount of sigma 32 to rise, so that heat shock responsive genes they may be. Other than this, there is the activity of repressors which can work towards heat shock response, and this also involves chaperones as well. So we'll take an example of Bacillus subtilis heat shock proteins, which are actually enhanced by a vegetative transcription factor. Here in this case, there is a far-off gene, HRCA gene, that forms a repressor, ORFA. Now this repressor can bind to an inverted repeat, that is a region of controlling inverted repeat of chaperone expression, that is CIRCE. Now chaperone helps in proper folding of this repressor so that it can bind to the operator region, and the heat shock operons are not expressed, so there is no transcription. In response to rise in temperature, there is continuous and aggravated unfolding of proteins. So now the chaperones move to attending these unfolded proteins, and as a result, the ORFA repressor is not properly folded, so it is no more available at the operator site, and in response to this, now the polymerase can bind and transcription can occur. So this is a very good example where the unfolded proteins, the chaperones and the repressors all are acting in response to a stimulus and guiding expression. Another example where proteases can affect the transcription of heat shock genes is in the SIG-E mediated regulation, where the transcription factor SIG-E is bound to the anti-sigma factor RSE-A localized at the membranes, a portion of it rising up the periplasm and a portion of it located within the cytoplasm. So after a heat shock, denatured outer membrane proteins, those will all activate a protease, that is DEG-S. Now this DEG-S can cleave within the periplasmic domain of RSEA. This is the first step of cleavage, which is followed by a second cleavage by RSE-P protease. So together they cause a cleavage in RSEA, the periplasmic region. Such region, it gets released into the cytoplasm along with SIG-E. Now this is sensed at higher temperatures by another protease, CLPXP, which further degrades the RSEA, and then the SIG-E gets released into the cytoplasm and now can become activated. Another level of regulation occurs through accumulation of LPS in the periplasm in response to the change in temperature, where the LPS gets recognized by sensor RSEB, which is located or associated with the RSEA. This RSEB, it gets dissociated, and this in fact destabilizes RSEA, which is now stepwise degraded, and the path follows, that is, it is released in the cytoplasm where it is acted upon by CLPXP and the SIG-E gets released. So here there are different examples of proteins, namely proteases, the chaperones and other sigma factors, which can sense the temperature. There is conformational change in them. They bring about a downstream signaling action and further can lead to adaptations occurring within the cells.